Next topic is membrane processes. So in the membrane processes, we are going to have ultrafiltration and reverse osmosis. Actually, the reverse osmosis is not a process used in the unit operations. So that is bioprocess technology. Only the ultrafiltration is going to be used. So this ultrafiltration is nothing but the filtration of the protein of interest or the molecule of interest. Okay, that is from the mixture of components by using a semi-permeable membrane. So that membrane is used to filter it. But this ultrafiltration and filtration, what is the difference means? Ultrafiltration is going to use the pressure so that the filtration may be very fast. At the same time, the methodology of the filtration can be modified in such a way to use the filtration process or the membranes in a reproducible way. Okay, that is we can use it for the multiple times instead of wasting in a time. Maybe we can use it for a number of times. Okay, this is called as ultrafiltration. But the life of the membrane is depends on what is the material, that is the raw material. What is the raw material we are going to use it for the filtration? If it is a slurry or something, it is having a lot of solid particles and maybe some other sedimentable materials that may clog the membrane. So before passing the material for the filtration, and you have to ensure that there is no sedimentable particles within the solution. If it is there, that will reduce the life of the membrane, and all the pores will be clogged, and automatically the cost of the operation may be very high. Usually this process is used to separate the high molecular weight one with the low molecular weight. So there is maybe low molecular weight proteins and high molecular weight proteins. For example, if you have your molecule of interest is 500 that is kd, or otherwise 50,000 kd, and maybe you can use such a way, that is means maybe the pore size is very specific. That means the 40,000 kd you can choose it, or 45,000 or 47,000 kd you can 47,000 Daltons you can use it, so that your protein will not penetrate into the pore size. That means within the pores it will not enter. So lesser than that is 47,000 or 49,000 or 50,000. So all the other components will be filtered off. So your protein and along with the same type of your proteins will be there, but this will reduce a molecular weight cutoff. So for example, your protein of interest is 50,000 Dalton, and you have to choose a filter size. The filter membrane size is 50,000 Dalton. So that pore size will not permeate or otherwise permit more than 50,000 or and above. So that means if your molecular weight is, if your protein of interest molecular weight is 50,000, so that is not filtered off, and the remaining particles, if it is 49,500 or 49,000, 40,000, 30,000, 20,000, including 200 Daltons, can be separated very easily. So there is a cutoff, molecular weight cutoff, to separate the proteins or the molecule of interest. So that is why this one is very famous. And it is not much time consuming, especially for the green that is green fluorescent protein separation. It is very famous for the separation, and this method is very common in the separation of the green fluorescent protein separation or purification process. We can have a look. There is a methodology of cross flow filtration, and that cross flow filtration that will enhance the life of the filter, the membrane. And so there is no further clogging or otherwise the blocking of the membrane components. The slurry depends. I already told the slurry is maybe if it is having more number of solid particles that may clog the membrane, and the nanofiltration membrane is very expensive. That is ultrafiltration membrane is very expensive. And it depends, maybe how perfectly you are using it, and how perfectly you are using the pressure, and if it is any spike in the pressure that may disturb or burst the membrane. So you have to be very careful, because this is time consuming, at the same time cost effective also. So I will play a video, okay, how it is separated, okay, for the purification of GFP, green fluorescent protein, okay, in the industry. We already had an experiment, okay, that is the cloning of GFP in the clinical laboratory, okay, in the practical 2. So this is the continuation of that one. So after cloning, we are going to grow the bacteria with the GFP gene, and after that we are going to purify it. How we are going to purify it, okay, this is the example.
We'll be using TFF for 2 different tasks within the purification process: concentration and diafiltration.
So you can see here there is a the concept is there, that is so here the green, this is green fluorescent protein. You can see the pore that will not allow this protein, but at the same time, okay, other proteins, other small proteins can enter into the pore and comes out. Okay, this is a membrane, and this is a membrane having the molecular weight cutoff. You can see the uniform molecular the pore size, that is a sheave. Okay, this will filter off, and automatically you will get only this type of molecules, and all the other molecules lesser than this size will be removed off.
Process: concentration and diafiltration. As the solution moves through the
So this is a method of the instrumental setup for GFP by using filtration, ultrafiltration. You can see here there are the supply tank, and waste will be there, and this is a pump will constantly pump it, okay, and you can maintain the pressure also. And this is the inlet of the filtration membrane and there are 2 the outlet, okay, that is one is GFP that is collected, okay, in the supply tank again, and another one is the waste one. Waste one means it is not your protein of interest, that is a waste actually, so that is collected as a permeate. Okay, this is a filtrate, I think. Just you can see the animation.
The TFF apparatus, it leaves the supply tank, is pulled through a pump, past a pressure sensor, and then across the filter membrane. Everything that passes through the membrane, including the buffer solution, is known as permeate, and for this process is sent to waste. The GFP protein is larger than the pores of the membrane and is retained. The retained material, called retentate, is recirculated to the supply tank. Recirculation of the feed continues until the desired concentration of GFP is achieved.
You can feed the retentate into the supply tank so that the continuously supplying, okay, this one is online mode, okay, there is a continuous mode, maybe permanently remove all the components other than, okay, this GFP. You can see here, so these are all the waste materials collected here, and to a particular extent automatically you will get only your retentate, and this outlet is going to be decreased. At that point you can stop okay, the filtration, ultrafiltration method. If there is no permeate is there, if there is no protein or outlet, okay, from here, maybe you can check the sample here, and if there is no delta A, that is absorption at 280 nanometer, automatically what you have to do, you have to stop the process and get the retentate and all the retentate into the supply tank, and you can collect it. That's all. So this is a very simplest method, and the only thing is, so this is the maybe the recurring one. You have to change it, and all the things are okay, nothing. Maybe you can use it continuously, maybe by using a complete sterilization procedure. Okay, maybe you can sterilize it and you can use it again. That's all. So this filter membrane is a little expensive part, but by using this method, and you can wash the properly by using, okay, this buffer, you can wash all the unbound or bound materials. Okay, again you can reuse this filter membrane, so before starting next procedure.
Following concentration, and while the protein solution is recirculating, a new storage buffer solution is added to the feed. In effect,
you can see here, this is a feed passed to the that is ultrafiltration.
The protein is being washed by the flow of a new buffer solution in and the old buffer solution out. As this diafiltration step proceeds, the buffer solution that's being added to the feed replaces the buffer solution that the GFP was.
The buffer solution addition is mandatory because to a particular extent the concentration of the protein will be more. At the time, okay, this may clog the membrane also sometimes. So you have to maintain the buffer, you have to add the buffer to the feed so that you can avoid the clogging of the membrane.
Originally in, effectively removing any remaining salt as well. When this process is complete, the GFP solution is
So this is ultrafiltration membrane component. Maybe if you have the RO filtration unit, okay, in your house, you can see that, okay, that is the same setup. But this is a larger scale. So that is a smaller scale, okay, for the drinking water purpose, and this is for maybe the large scale, including UF membrane based filtration or water purification systems are available now like Kent or AquaGuard and all. Okay, maybe at the time of dismantling and assembling or refilling the cartridges, you can ask the person, okay, to show the UF membrane cartridges also. So that is very important, and that is a small one, okay, in the drinking water supply. Okay, this is the biggest one. The different layers will be there, and simultaneously you can purify it at a faster rate.
Is routed through a 0.22 micron final filter and then collected in appropriate containers, usually bottles or bags. The purification process.
Finally, the output will be passed through the 0.2 micron filter to filter any the contaminants like any bacteria or something. So you can filter it off and remove all the bacteria or maybe any pyrogens, okay, from the protein solution, and finally you can collect GFP protein. Okay, this is outlet and this is the final output. You can collect it.
Is complete. The green fluorescent protein concentrate can now move downstream to final fill finish to be freeze-dried and packaged.
Now this is the feed for other type of purification. Okay, this is UF ultrafiltration is not a last stage. You should remember that ultrafiltration should be the starting, or otherwise the next to the ammonium sulfate precipitation, or sometimes, okay, without doing ammonium sulfate precipitation, like this type of protein you can do this one, and the next level it can be added directly to the GFP, that is chromatography using gel filtration chromatography. That is GFP directly poured into the gel filtration chromatography, and you can get the purified fractions in the final rate. So this is the procedure of protein purification using ultrafiltration. Next, coming to the criteria of purity by SDS page method. You already studied, or otherwise maybe in the first year, the first semester practicals, maybe you had an idea about what is SDS page. That is sodium dodecyl sulfate polyacrylamide gelatophoresis. Now we already completed all the process of purification, starting from ammonium sulfate precipitation, and after the dialysis, and after that Ionic exchange chromatography, gel filtration chromatography, and after that affinity chromatography, and if possible, okay, you can go with that is hydrophobic inclusion chromatography or reverse phase chromatography and all, and finally you will get the purified product. Now it is the time to evaluate whether your product in your hand, that is the purified one, is purified or not. How many fold it is purified from the stage, each and every stage. For example, if you have the culture filtrate, so that is the starting material, so that is the one you are going to purify it. So next one is ammonium sulfate filtration, or otherwise ammonium sulfate precipitation. Next one is ion exchange, or otherwise gel filtration, and last one is affinity chromatography. And each and every stage, how much amount of purification occurred, and what is the yield of your purification, that we are going to study. Students, this is a very, very important one, and if you are going to the industry, okay, this is a mandatory thing, and lot of questions will be asked, okay, at the time of interview. Just I'll show the animation of SDS page, just to have a look.
SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis, is an electrophoresis method and very powerful technique in the study of proteins. In SDS page, proteins are separated in a polyacrylamide gel based on their molecular weight. For sample preparation, a loading buffer containing SDS beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein samples. Protein samples may be biologically derived, for example from prokaryotic or eukaryotic cells, tissues, viruses, environmental samples, or purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain. Proteins are formed by linking amino acids with peptide bonds and fold into one or more specific spatial conformations, driven by a number of interactions such as hydrogen bonds, hydrophobic interaction, disulfide bonds, and ionic bonds. SDS is an anionic surfactant and contain a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions, while the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, when a protein mixture is heated, SDS binds uniformly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by SDS. This treatment brings the folded proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. For gel preparation, a separating gel solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate and T-med. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first. The polymerization reaction is a vinyl addition polymerization initiated by a free radical generating system. Ammonium persulfate is used as the free radical initiator, while T-med accelerates the rate of formation of free radicals from persulfate. These free radicals convert acrylamide monomers to free radicals which react with unactivated monomers to begin the polymerization chain reaction. The elongating polymer chains are randomly cross-linked by bisacrylamide, resulting in a gel with a characteristic porosity. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight samples. The stacking gel solution is poured on top of the solid separating gel, and a plastic comb placed on the top of the stacking gel during polymerization enables the formation of small wells in the gel. Once the gel completely polymerizes, the comb is removed, and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel. The gel is inserted into a chamber, and a tris-glycine chloride buffer system with pH 8.3 is poured to allow the conduction of current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample application, in addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes and thereby allows the estimation of the sizes of the proteins in the actual samples, which migrate in parallel in different tracks of the gel. Each sample is pipetted into its own well in the gel. And as we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, and glycerol increases the density of a sample and used to make the sample fall to the bottom of the sample well, rather than just flow out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel. The stacking gel is used to ensure that all of the proteins arrive at the separating gel at the same time. Because gel wells are around 1 cm deep, so in the absence of a stacking gel, proteins would all enter the separating gel at different times, resulting in very smeared bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So, in the sample wells we will have proteins, chloride ions, and glycine. Proteins and chloride ions are negatively charged. Well, glycine can exist in 3 different charge states depending on the pH. The isoelectric point or pHi of glycine is equal 5.97. At this pH glycine carries neutral charge due to the gain and loss of protons. At a pH below its pHi, glycine carries a net positive charge. And at a pH above its pHi, it becomes negatively charged. The running buffer pH is equal 8.3, which above the pHi of glycine. So, at this pH glycine is negatively charged. Once the electric current is applied, proteins, chloride ions and glycine are forced to enter the stacking gel, where the pH is equal 6.8. At this pH glycine switches predominantly to the zwitterionic neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions on the other hand, move much more quickly. Chloride ions migrate in front of the proteins as leading ions, and glycine molecules migrate behind the proteins as initial trailing ions. Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ions and glycine. This procession carries on until it hits the separating gel, where the pH switches to 8.8. At this pH the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecular weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins. Due to the relatively small molecule size of bromophenol blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the dye and before the samples have completely migrated through the gel and leave it. At the end of the electrophoretic separation, all proteins are sorted by size and can then be analyzed by other methods, for example protein staining methods and immunological detection, such as the Western blot technique. After removing the glass plates, the stacking gel is discarded and the separating gel can be stained. Kamasi blue is the most popular protein stain. It is an anionic dye, which non-specifically binds to proteins. The structure of Kamasi blue is predominantly non-polar, and it is usually used in methanolic solution acidified with acetic acid. Proteins in the gel are fixed by acetic acid and simultaneously stained. After staining, the proteins are detected as blue bands on a clear background. The molecular weight size markers in a separate lane in the gel can be used to determine the approximate molecular mass of unknown biomolecules by comparing the distance traveled relative to the marker.
Yeah, this is one of the best animation for SDS page. I hope you can understand the concept of SDS page. Now we are going to the criteria of purity. Yeah, this is the output. Just now you had an idea about what is the staining method, but here in protein purification and all, and we won't recommend the the Kumasi brilliant blue staining method. Here we have to use only silver staining because that is more sensitive than Kumasi brilliant blue, and this is a little complex at the same time. This is more sensitive, that is why we are going to prefer that one. And if you are not familiar with silver staining, you can use Kumasi brilliant blue. That is non-specific binding, and maybe in the industry and all, for the confirmation of criteria of purity, we have to use only SDS page with silver staining. You can see here this is the band number 1, and this is 2, 3, 4, 5 and 6. Usually the first band and the last band usually the standard protein markers, and we already explained in the animation the standard or known proteins. These are all the different protein standard markers will be there, and so this is the first maybe the sample, that is the second lane 2. You can see here lane 2 is your crude extract. That means either the culture filtrate, you can take that one. You can see how many number of proteins are present in your crude extract. So finally you're going to purify your own protein, your protein of interest. Okay, this one you can see, okay, this band is available here, and this also. These are the different stages, and finally this is the last method for your protein purification, and you will get only this type of homogeneous protein. That is, the protein is 100%, not exactly 100%, exactly 97 to 98% purity. Okay, and lane 3 is ammonium sulfate precipitate. You can see here, okay, this type of fractions are removed by ammonium sulfate, and including this region, and this region is also removed by ammonium sulfate precipitation. You can see here ion exchange chromatography. This is the chromatography, and ion exchange chromatography removes a majority of the areas. Okay, this area and this area and this one. Okay, these type of proteins are removed, and this protein is also removed, and so some of the traces, like, okay, this one is also removed here. Okay, this is ion exchange chromatography. And finally gel filtration chromatography, that is GFP. That is gel filtration chromatography is a very important. The gel filtration method is used, and you can get only this type of fraction, and that is collected in a particular the fraction collector. So this is not a copy of something, okay, this photo from internet or something. Okay, this is from my own purification, and this is the purification of acetylcholinesterase enzyme from human brain. Okay, this is ammonium sulfate precipitate. Okay, the crude extract ammonium sulfate precipitate. This crude extract means, okay, this is extract of the cortex, that is a human cerebral cortex, okay, extracted with the homogenate, and this extract is filtered, and this filtrate is called as crude extract. And the next one is ammonium sulfate precipitate. I took 60 to 62%, and exactly 60% fraction is having the enzyme fraction, so that is why I took it. Okay, that is the one. The 60% is having, okay, this much of proteins. And next one is ion exchange chromatography, that is the DA cellulose chromatography. So this chromatography, that particular active fractions are pooled, and active fractions giving, okay, this many bands. And finally we have to go for gel filtration chromatography, and I got this fraction as a single band. So this is the beauty of the protein purification. Finally you will purify your protein of interest, and so that is a big achievement. So now we are coming to the quantification of your purification, that is called as so-called protein purification table. Okay, and the construction of the protein purification table is very, very important. And if you are a protein technologist or protein bioprocess technologist, and you should know about, okay, the construction of the protein purification table, and we can have, okay, that one. These are all the different parameters at present. Okay, 8 parameters are there. First one is the volume of the protein or enzyme solution, the starting material, okay, what is the volume, and the protein content in the solution in 1 ml, okay, how many milligrams are there. You can do the protein estimation by UV method or Bradford method or Lowry method, and you already did Lowry et al method and Bradford method, okay, in your UG and PG level. Okay, this is a very simple method. And UV method without adding any reagents, that is directly using 280 nanometers. Okay, that is very best method for protein quantification. The activity of the enzyme solution may be based on your enzyme. For example, if it is a phosphatase enzyme, you have to use the phosphatase substrates. Okay, and so this, for example, if my enzyme is cholinesterase, means, okay, I have to use the protocol Hellman's method, okay, for determining the activity. So for each and every enzyme, this activity determination is called as assay. Assay means quantification of enzyme activity that will vary for each and every enzyme. And total amount of the enzyme is activity in particular volume. For example, if you have 15 ml of the fraction, you got it, okay, in the each and every stage, means in 15 ml how much amount of activity in units per ml, that you have to calculate it, okay, the total amount of enzyme. And specific activity means units, okay, the total amount of units of international units of enzyme in milligram of the protein. For example, if you have 50, for example, this, if you have 50 ml, in 50 ml how many units are present. At the same time, in 50 ml how much amount of the protein is present in the milligram. Okay, in 50 ml how much of activity, okay, that is activity divided by the protein content. So again, the protein content can be derived by 3 methods: UV, Bradford, Lowry et al methods. And nowadays, okay, the BCA method is very famous, and if you have the fund, you can get the BCA chemicals for the by picolinic acid, and that method is very sensitive also. But only one problem is, okay, that reagent is a little expensive. Okay, and the next one is the yield. How much yield is there? Okay, maybe for each and every step, how much the yield percentage. That means how much of the percentage you purified for each and every step. Last one is the purification factor. How many fold purification you did it? 40%, 30%, and what is the number of fold? How many fold you purified it? Okay, that is a very important one. Just you can have a look. Okay, this is the protein purification table of my enzyme, and I gave, okay, the values I got it. Okay, to give the explanation to you very easily. And the first one is the crude extract one. The CE represented as a crude extract. I told you, okay, this is a brain sample is my sample, and the human brain cortex, okay, there is a cerebral cortex, okay, is taken and sliced, and I got completely the homogenized and again filtered off, okay, and centrifuged it. The clear supernatant containing okay, the clear solution, okay, that is called as crude extract. Okay, that is the starting material of my purification. The total volume is 500 ml, and the total activity is approximately 3,000 units are present. International units of enzyme activity is present, and I checked 1 ml how much amount of milligram, and I calculated for 3,000 ml. So 500 ml, and this is 15,000 milligram is present. That means in 500 ml of my starting solution, I have 3000 IU, that is international units of enzyme. At the same time, the same 500 ml having 15,000 milligrams. Okay, so of proteins are present. So next one is coming to the specific activity. Specific activity I told, okay, in the previous slide, okay, that is activity divided by protein content. So you have to check the activity is already there, and you have the protein volume is out there. So 3000 divided by 15,000 is the value, that is 0.2, and so this is called as specific activity. And maybe the next one is the yield, percentage of yield. So this is the starting material. Actually, this is starting material means this is having 100% of the enzyme. That's why. So always the first one is 100%, and there is no purification here. That is why you can put dash, because there is no step is involved. So this is starting material. Second one is ammonium sulfate fractionation. So this is AS means ammonium sulfate fractionation. I used 60% and added for sequential ammonium sulfate precipitation 20%, 40%, 60%, 80%, 100%, and I proved that, okay, 60% is the best the concentration of ammonium sulfate to precipitate my protein. So this ammonium sulfate precipitate is reconstituted with the phosphate buffer and cold phosphate buffer, that is 20 millimolar, and this one is made up to 100 ml, and this is already dialyzed. You know very well, okay, what is the meaning of dialysis. I already explained, okay, what is the meaning of dialysis and how to do the dialysis also. So this is 100 ml. So the total volume is 100 ml now. So from 500 ml, and I precipitated the proteins of my interest, okay, by 60% ammonium sulfate, and after that I dialyzed it and removed all the ammonium sulfate also, and after that I got 100 ml. So this is the starting material for the second step. In 100 ml, what is the activity? I checked it, okay, all the assay, and the enzyme assay showed 2400 IU is present, and I checked the protein concentration in 100 ml, that is 4000 milligrams. You can see the drastic decrease in the protein concentration. At the same time, there is no much difference in the enzyme activity. If there is a huge loss of enzyme activity, that may be a problem. You have to ensure that your protocol is wrong, or otherwise you made some mistake, or otherwise your handling or the physiochemical properties of handling the enzyme is bad, because enzymes are very sensitive. Because if you are keeping the enzymes in the room temperature, your enzyme activity will be lost. The protein will be there, but enzyme activity will not be there. Okay, so this is the protein concentration. The 2400 divided by 4000 is specific activity is 0.6, and the yield is okay, here. Coming to the yield, so what is the total yield of this step? That is ammonium sulfate. So this is the first step of purification, and this is the second step of purification, and this is a third step of purification. This is not a purification step, this is starting material. Okay, so that is why there is no purification factor, and all the 100% of your enzyme is present here. So from the 100%, how much you got it for the second step? Okay, that is approximately 80%. How you got 80%? That means you have 2400 IU, okay, and so 2400 IU divided by 3000. That means starting specific activity. So the calculation of this unit is there. That is 3000 units are present. So this 3000 and here 2400. So here 2400 IU divided by 3000 IU. So multiplied by 100, because this is percentage. That means multiplied by 100. Okay, that is giving 80. So this is 80%. So from this step to this step, and you got the yield from 100% of the enzyme from 500 ml from the culture filtrate. Okay, that is a crude extract. You got 80%. That means 20% you lost it. Okay, so that means the loss is inevitable. That means for each and every step the loss will be there. Okay, for the particular step you cannot purify 100% exactly. Some loss will be there. So this is a reasonable loss, so there is no issues. And maybe the next step, okay, this is a calculation of purification factor. So for the yield percentage you have to calculate only the total activity, and for the purification factor you have to consider only the total protein concentration. Okay, that is a specific activity related one. That means unit divided by milligram is specific activity. So this is activity you have to consider for yield, and the specific activity has to be considered for purification factor. You can see here for the first step of ammonium sulfate precipitation, the 0.6 you are getting. Okay, 0.6 is the specific activity. So 0.6 divided by 0.2, okay, that is the value 3. So purification factor is, that is fold of purification is 3 times you folded. Okay, maybe you 3 times fold purification you achieved. Okay, that is the main thing. And second is IEC. That is, IEC means ion exchange chromatography. You can see here IEC is ion exchange chromatography. And during the chromatography I got a few fractions of my purified enzyme, and I pooled all the fractions, and active fractions are pooled, and this is only 45 ml I got it. So this 45 ml I got, I checked the activity, that is the 1440 IU, and in the 45 ml the total protein concentration is 500 milligram of protein is present. So I easily calculated the specific activity is simple. Okay, that is 1440 divided by 500 ml. So 500 milligram of the protein. So this is the value of the specific activity. And from total enzyme activity, that is IU, I calculated the yield, that is 1440 divided by 3000. Starting is 3000 IU, so that is why 1440. In this step 1440 you have, and the starting is 3000, that is why 1440 divided by 3000 multiplied by 100, because it is a percentage, that is why you will get 48% of purification. You can see from 80% to 48% you got it. And coming to the protein purification factor, okay, I already told, okay, this one is you have to consider only the total activity, and this one you have to consider only the specific activity. You can see here the second step you got it 0.6, that is by 0.6 divided by 0.2. But in the third step you got 2.9. So 2.9 is the present step, and for the first step you got 0.2, that is by 2.9 divided by 0.2, that is 14.5 is the protein purification factor or fold purification. Okay, that is the one. Next, coming to the last one, that is gel filtration chromatography, GFC, and this GFC I got 50 ml concentration. You can ask the question why this volume is increased, because always GF you will get different fractions and the fraction volume may be more, and this I got 50 ml. And this 50 ml active fractions are pooled, and all the total volume is 50 ml, and the total activity is 1000 IU is there, and the total protein concentration is 125. You can see here in 500 milligram I got 1440, in 125 milligram I got 1000. You can see the difference of purification fold. Okay, and this is specific activity, that is how to calculate the specific activity, this IU divided by milligram. Okay, that is 1000 divided by 125, that is 8. Okay, that is a specific activity. And coming to the yield percentage, the yield percentage is the calculation similar to the method. Okay, that is you got here 1000 is for For yield percentage, you have to calculate only the total activity. For protein purification factor, you have to consider only the specific activity. Both activities you can consider. Okay, the first one is total activity. Here 1000 and the starting material is 3000. 1000 divided by 3000 multiplied by 100. Okay, that is 33%. And next one is purification factor. Purification factor is a specific activity, that is 8 divided by 0.2. Okay, that is why it is called as the purification factor 40. So 40 times you did the purification, that is the purification fold or purification factor is 40. So 40 is a decent or otherwise standard purification step, and if you are able to get 40%, and maybe you are a typical protein purification specialist. And this method is the actual values I gave, okay, because I got it from my research notebook and gave it to you to understand the concept very clearly. And this is the meaning of protein purification table, and you have to construct, okay, this protein purification table and all the steps. Okay, these are all the steps involved, and these steps, okay, may vary for you. Maybe if you are doing the culture, maybe crude extract and ammonium sulfate precipitation and ion exchange chromatography 1, 2 and affinity chromatography may be another step. So the each and every step may vary for each and every person and each and every process of purification. And the second column, all these column will be there. Okay, there is volume, total volume of starting material and total volume of activity, total activity in IU, total protein in milligram, specific activity units per divided by milligram, and yield percentages, the calculating the value of total activity of the particular step but divided by the starting step. And purification factor, you have to calculate specific activity, that is the particular step, and from the starting step. So you can see the starting step is 0.2, 0.2, 0.2, because this is the starting one, and you can see here this is 3000, 3000, 3000, that is the starting one is 3000 here. Okay, I hope you can understand, okay, the concept, and this is very important, and make a note of it. So with this I will conclude the protein purification steps for the bioprocess technology. Thank you.